All right. So again, we're up here on this peak uh, where we know about the basics of what's now called molecular biology. We now understand the way that heredity works. We went from everything from Mendel's discovery of genes to microscopists' discoveries of chromosomes, to Thomas Hunt Morgan's and company's realizations that the two are one and the same, that genes are physically carried on chromosomes, to the discovery that chromosomes were made of DNA, to the discovery of DNA's double helix shape, and the discovery that the order that the nucleotides appear in DNA carries information. That information is used to make pieces of messenger RNA in a process called transcription. And the messenger RNA is used to direct the assembly of proteins out of amino acids by the process of translation. So that's how we got from base camp all the way to the top up here. And we've talked about viruses as something that we can now understand far better now that we have the knowledge that we do. And something else that's been in the news an awful lot that I feel almost obligated to talk about is the human immune system. This is the way that we defend against pathogens. A pathogen is anything that makes you sick. It includes viruses, it includes bacteria, it might also include fungi or even protists. These are agents that cause disease. And your immune system is the system in an animal's body. I'll be talking almost entirely about the human immune system, but other organisms have the same or at least similar systems. This is the way that you defend against pathogens. And if you're born without an immune system, like unfortunate David Vetter shown right here, you have to grow up in a sterile plastic bubble because any bacterium, any virus could cause a life-threatening infection. And we can talk about your immune system as having several parts, and one is the innate immune system. And the innate immune system works against anything, and it doesn't require sensitization. If we think of the immune system as being like a defending army, the innate immune system would be equivalent to things like the trenches and the barbed wire, and maybe, fortress walls and landmines and things like that. That are just permanently there and will stop anything that tries to get too close. So these would include things like anatomical barriers, especially your skin, inflammation and fever, so-called white blood cells patrolling your body called leukocytes, and a group of specialized proteins in your blood called the complement protein system. So if we look at these, anatomical barriers include things like your skin, which is tough, is thick, and as you see, it's peeling right here. Your skin is constantly shedding its outermost layers. There are actually bacteria that are adapted to live on your skin. In fact, everybody has well over 100 different bacterial species living on your skin. And indeed, if you get too many of them in certain parts of your skin, you start smelling bad. Bacteria are the cause of things like underarm odor. But since your skin is constantly shedding its outermost layers, it's harder for pathogenic bacteria, disease-causing bacteria, to gain a foothold. Of course, they can, if your skin is broken, bacteria can get in. If you've ever had an infected cut, you know what I'm talking about. Most bacteria in your food can be broken down by your stomach acid and your stomach's digestive enzymes. Uh, is the skin just a part of the innate part of the immune system? The skin's actually considered an organ. I've seen it referred to as the integumentary system, but it certainly plays an immune system role. We can treat it as a contributor to the anatomical defenses of the immune system for our purposes here. Okay, so digestive acids break down bacteria in your food, or most of them anyway. There are some that can get through, which is how you get food poisoning, but most can be stopped in your stomach. You don't get more infections in your eyes because your tears contain an enzyme that breaks bacteria down, an enzyme called lysozyme. Lyso is Greek for to break apart or to dissolve, and lysozyme breaks down lots of things, but in this case, it breaks down eye bacteria. You also have the mucus, the phlegm in your respiratory system, your lungs and your bronchi and all that, which trap bacteria. And since mucus is always being pushed out, is ultimately coughed up and then either swallowed or possibly spit on the sidewalk. Either way, coughing up and getting rid of mucus can get bacteria out of the lungs before they get a chance to grow. So that mucus can be part of your innate system. Anytime you've got cells that get damaged, they send out signal molecules. We don't need to worry about what those are, but those molecules trigger the inflammatory response or inflammation. The ancient Roman author Galen, one of the first great medical authors, called it rubor et calor et tumor et dolor, which is Latin for redness and heat and swelling and pain. Excuse me. There, hopefully got rid of some pathogens there. The redness and the heat are caused by increased blood flow. For reasons I'll talk about in just a minute, blood vessels get increasingly leaky. Actual blood does not normally get out of them, but fluid can pass from the blood into the inflamed tissues, and that causes swelling. And there's also increased sensitivity, which causes pain. So redness, heat, swelling, and pain is the perfect definition of inflammation. And again, this is something you have no doubt experienced if you've ever had an infected cut or an infected ingrown toenail or something like that. It doesn't even have to be caused by infection. Any kind of cell damage, you'll see this. So you'd see this in something like a burn as well. And then fever is also part of your immune response. And whereas we used to treat infections by trying to bring the fever down, mild fever is not particularly bad for you. In fact, it's good. Many pathogens can only survive within a narrow range of temperatures. So bacteria in particular can be slowed down if the temperature is too hot for them. Obviously, the fever does need to be brought down if it's so high that you're risking damage to the body itself. Think of any fever over 103 Fahrenheit, which is what, 41 Celsius, is potentially dangerous. But mild fever actually seems to enhance other immune defenses as well as slowing the bacteria down. So it's not necessarily a bad thing to have a fever. Inflammation also triggers a population of cells in the body called white blood cells, or leukocytes. Leuko is Greek for white, so these are your white cells. These travel through your bloodstream, but they're also capable of forcing their way out of your blood vessels and traveling just about anywhere in the body. In particular, they can get to the infection site because of those leaky blood vessels that we talked about earlier. They can engulf bacteria. They basically attach to a bacterium and kind of flow around it and end up completely surrounding it and breaking it down because they have lots of lysozyme, this all-purpose digestive enzyme that breaks things down. They can also engulf debris from broken cells. They can go after anything that's needed. And if you have very large numbers of leukocytes around an area of bacterial infection, if you have enough of them all together, they basically make up this creamy goo known as pus. Yeah, sorry, but pus is basically masses of leukocytes and broken down cells and broken bacteria and things like that. As for what makes white blood cells, they're made in your bone marrow. They're also made in an organ called your spleen. You can live without your spleen. Spleens sometimes have to be removed because they bleed a lot if you have trauma. People in car accidents sometimes have to have their spleens removed. And the bone marrow can usually take over, but your immune system is not quite as effective as it was if you have to get your spleen taken out. Okay, finally, your liver makes a huge number of different proteins. Your liver makes the proteins that make it possible for your blood to clot. Your liver makes a lot of enzymes that break down various products in your food, such as alcohol that we talked about. And your liver makes over 30 specialized proteins that are called the complement system. And some of these complement proteins, called the membrane attack complex, can quite literally punch holes in pathogenic cells. Here you've got a diagram of proteins that are numbered C6, C5b, C7, C8, and then a bunch of C9s. These proteins attach to each other one by one, and once the C9s are in place, they open up a hole into a bacterium, let's say, and blow it open. Right? So these are like, I don't know, like, these are your anti-tank rockets, I guess. Complement proteins also attract leukocytes and stimulate them to engulf bacteria and broken cell bits and things like that. So all of these are parts of the innate system. They don't have to be triggered. They're always there, and they are always effective. Just like if you've got a building you don't want anybody to get into, you can put some barbed wire around it and then pretty much leave it alone. That barbed wire should always be there and always be effective against anybody that's trying to get too close. But your adaptive immune system has to be triggered. It has to be stimulated by something. What's more, it can be tailored to respond to specific pathogens. Whoops, sorry. Okay, sorry, I was checking the chat. All right. Your adaptive immune system can be tailored to respond to only particular bacteria or viruses or what have you. And it can remember specific pathogens that you've been exposed to for years or even decades. And if the same pathogen ever invades again, the adaptive immune system can gear up very quickly and stop the pathogen before you're even suffering symptoms. So yeah, the immune system includes this component that has a sort of memory associated with it. Not a conscious memory, but the ability to retain information about everything you've previously been exposed to and use that if you ever get exposed to it again. There's a second type of white blood cell I didn't mention. We talked about leukocytes. By the way, I don't know if you've ever had this done or seen it done, but doctors will sometimes order something called a complete blood count. They'll take a blood sample and the tech will smear it out on a slide, and there are ways of counting the number of white blood cells that are present. And we're not going to go into this because of time, but there are different types of white blood cell that you can tell apart under the microscope. And you'll find some if you have a bacterial infection, and other types if you've got a parasite. So you can tell a bit about what kind of infection you've got by counting the number and types of leukocyte. But there's another type of white blood cell called a macrophage. And you have yet a third type of cell called dendritic cells. And in this computer graphic, this dendritic cell is the thing that looks kind of like a purple carnation down below. What they can do is pick up fragments of whatever is triggering your immune system, fragments of dead bacteria, pieces of virus capsid, bits and pieces of a parasite infecting you, whatever it might be. They can pick up fragments or even just large molecules from whatever has got your immune system stimulated. And incidentally, anything that's triggering your immune system is called an antigen. And the macrophages and dendritic cells can physically pick up pieces of your pathogen, pick up antigens, and present them to yet another cell population called T lymphocytes. There's your T lymphocyte is shown up there in blue. T lymphocytes are mostly found in a system of structures that you may not really be aware of, called your lymphatic system. Your lymphatic system is a system of very thin-walled ducts that passes through most of your body. Lymphatic vessels are connected to lumps of specialized tissue called lymph nodes. And you might, if you've ever had a throat infection, if you remember, if the doctor kind of feels you around the angle of your jaw, right, kind of where your jaw meets your neck, what he's checking for is swollen lymph nodes. Lymph nodes are these lumps of tissue that are connected to each other by lymphatic vessels. And the whole thing makes up your lymphatic system. And lymph nodes tend to enlarge when you have a nearby infection. Why do they enlarge? Well, they're full of T lymphocytes, and there are millions of different types of T lymphocytes. And I wish I had more time to go into this because this is really cool. But when your T lymphocytes are being formed, or the cells that become your T lymphocytes, when you're still an embryo, something happens that doesn't happen anywhere else. In almost all of your other cells, almost all of your cells have the same DNA that the fertilized egg that you came from had. What happens is that different types of cells, the DNA that they're not going to need gets shut down. So your liver cells still have the DNA for making muscle proteins, but those genes are turned off in your liver cells and they're not normally used because your liver doesn't have to make muscle proteins, it has to make liver proteins. So different types of cells have different genes that are on or off, depending on what type of cells they are. But almost every cell in your body has the same DNA that the fertilized egg that you came from originally had. Just different parts of it are switched on or switched off. The difference is in your T lymphocyte, because as your T lymphocytes are made, the genes that code for receptor proteins are spliced and randomly shuffled. And so you end up with millions of different types of T lymphocytes each one of which has differently shaped receptor proteins on its surface, because each of those types has different genes, because those different genes have undergone this process of splicing and shuffling. So this is an exception to the rule that almost all of your cells have the same DNA, just different parts of it active or not. And that's a computer model right there of what one of these receptor proteins looks like. And they sit on the surface of the cell. And the macrophage or the dendritic cell goes around to the T cells in the nearest lymph node. And if it can find a T cell whose receptors just happen to fit the antigens that it's carrying, the T cell is stimulated to divide. Now, this diagram probably looks like kind of a mess, so let me break it down. I'll talk more about this later, but this purple and orange sheet-looking thing that's up here, that's the structure of a cell membrane. I will talk a little bit later as to why it looks like a couple of shag carpets. But that's the membrane. Up at the top, that's the outer membrane of the macrophage or the dendritic cell. It's called APC, that stands for antigen presenting cell. And stuck onto the outer membrane is this thing in orange, and that is some kind of fragment of whatever pathogen is making you sick. So the macrophage is carrying this thing on the outside, and it goes around in your lymph node looking for a T cell whose receptor protein, which is shown here in blue, just happens to have a shape that fits the antigen. It's like you've got a key and you go around from door to door trying to figure out what lock might fit it. And the antigen presenting cell should eventually find a T cell whose receptors fit the antigen. Just like if you have a key and you try it in every lock, eventually you'll find one that opens. And we see, again, this is another really important example of how the shape of proteins determines what they can do. And so if the antigen matches the T cell receptor, that T cell is triggered to divide. And that T cell gives rise to two different populations of cells, cytotoxic Ts and helper Ts. Cytotoxic Ts, otherwise known as CD8 cells, those are what go after virus-infected cells. If you've got a virus, that virus is replicating in your own cells, and your CD8 cells start patrolling for virus-infected cells, and they cause those virus-infected cells to commit suicide before they can release the viruses. Hence the name killer Ts. Cytotoxic T cells also go after cancer cells as well. Your body does have natural defenses against cancer, although all too often that's not enough. A T cell may also give rise to helper Ts. We don't really need to know how they do it, but they can stimulate the immune response in various other ways. Incidentally, if your helper T cells are infected with the HIV virus, your immune system can't respond as effectively, and you may have a case of full-blown AIDS, immune deficiency disorder, if your CD4s get knocked out. Okay, but there's another population of lymphocytes called B cells. The T cells can handle, well, the helper T cells are kind of cheerleaders for the whole process. The cytotoxic T cells will go after viruses and go after cancer cells. The B cells pick up antigens by themselves. They don't need those macrophages to present them with antigens. And again, there's many millions of different types of B cells. Each one has different receptor proteins on the outside. And those millions of different types are created by certain genes that are randomly spliced and shuffled. So every B cell is genetically different from every other B cell. And a B cell will, hopefully, if it's presented with an antigen that matches its receptor type, that B cell will be stimulated to start dividing like crazy. And the B cells become a type of cells called plasma cells. And the plasma cells give off proteins called antibodies. And that's what an antibody looks like. They're kind of Y-shaped proteins. And the antibodies will have the right shape to fit the antigens. Every B cell has a different type of receptor, and every B cell can make antibodies that have the same shape as that receptor. So every B cell can make a slightly different type of antibody. And when you have an infection, bits of whatever is making you sick, bits of that pathogen, if they find their way to the B cells, and the B cell that happens to have the right receptors picks up that antigen, that B cell will start cranking out antibodies. And the antibodies physically stick to antigens. They physically bind to them. Again, that's all a function of protein shape. Antibodies can link them together so that instead of being able to move freely, bacteria start getting clumped. Bacteria start getting stuck together. This is a virus in the center with spike proteins. This could well be COVID. And the antibodies are the things that are surrounding it that are attaching to its spike proteins, blocking them from working. And you're not seeing it in this picture, but they might cause virus particles to clump together, which tend to slow them down. Antibodies trigger the complement system that we talked about that can blow a hole in pathogens. And antibodies also stimulate white blood cells, leukocytes, to devour whatever they're attached to. Antibodies stuck to a bacterium make that bacterium, quote, especially tasty for white blood cells to engulf. By the way, I use the term phagocytes right here. By that, I mean white blood cells or leukocytes. Phagain is Greek for to eat. So phagocytes are eating cells. In this case, that refers to leukocytes, the white blood cells. And yeah, antibodies encourage or stimulate leukocytes to engulf whatever it is they're attached to. It's a process called opsonization. Opso is Greek for a feast. And the white blood cells are encouraged to feast on whatever the antibodies are stuck to. You might recall President Trump had this, and some other people have. One therapy for COVID infection is you can take antibodies from a person who's already had the disease and inject them into a sick person. That sometimes works in very severe cases. There's a whole technique that I'm just not going to have time to go into, where in the lab, you can induce rabbits, for example, to produce antibodies that are targeted against a very specific pathogen. They're called monoclonal antibodies. And this is another one of the treatments that is available for COVID infection, is you can get injections of lab-produced monoclonal antibodies that specifically go after the virus. And if you've been following progress in virus treatment, you might know that this is an option that's out there. It's a lot more expensive than the vaccine, but it does seem to work. at least in very severe cases. You can boost your own immune system by adding extra antibodies. But it sure might be nice if you could make your own antibodies before you even got infected in the first place. So how do we do that? Some of both the T cells and the B cells become a special cell type called memory cells. These are samples of mouse intestinal tissues, and the memory cells have been stained red. And the memory cells are just patrolling, passing through all parts of the body, just talking on their cellular phones and, doing their thing. And if the body is ever challenged with an antigen that it has already been exposed to, the memory cells can very quickly respond. They can quickly divide, and they can produce new cytotoxic Ts, helper Ts, and or plasma cells. So as long as you've got a few memory cells, you can very quickly mount an immune response against any pathogen that you've already been exposed to. This is why most people only get chickenpox once. I'm not sure that immunity always lasts a lifetime, but it does last a long time, that once you've had it, you don't get it again, because if that virus is ever encountered again, your memory cells can transform into cytotoxic Ts and attack and kill virus-infected cells before the virus has much of a chance to even make you sick. And there's other viral diseases like measles, I think, or mumps, where once you've got them, you are not supposed to be able to get them again, or at least not for a long time. Sorry, I've got a cat crawling on me. And incidentally, thanks to memory cells, when a woman begins lactating, she starts producing antibodies to all the pathogens that she has ever been exposed to. And human breast milk is pumped absolutely freaking full of antibodies. There's more in the breast milk than there are in the mother's own body. And newborn babies are still kind of fragile. Their immune system is not completely up and running yet. But as long as the baby's breastfeeding, that baby's digestive system is going to be protected by the mother's own antibodies against everything that the mother herself has ever been exposed to. I'm not in the business of telling people how they ought to take care of their kids, even if they have them. But this is one advantage that breast milk does have that we can't replicate with formula. That you do give a breastfeeding baby does get protection from the mother's own antibodies. And this is also the idea behind vaccines. If you give the immune system viruses or bacteria that have been broken up or killed or weakened, the immune system will respond to those killed, weakened, or broken up pathogens as if they were the real thing. The immune system can't tell the difference. So the immune system will quickly break down your broken or weakened or dead viruses, bacteria, etc. You may get a little bit of inflammation. When you get a vaccine, it often leaves you sore for a bit. But the idea is that memory cells can last for years afterwards, possibly for a lifetime, although these days with a lot of vaccines, you do need to get periodic boosters. But they will at least last for many years. And that means that since your immune system has been sensitized by a killed or weakened or broken virus, bacterium, etc., then if the real thing ever attacks, the immune system can respond very quickly. And that's the idea behind vaccines. The Pfizer and Moderna vaccines against COVID consist of messenger RNA. And now we can finally talk about this because you should know what messenger RNA is. This was a vaccine technology that began to be developed in the 90s. There was a lot of talk about how these vaccines are completely untested, but we've known the basics of how to make a messenger RNA vaccine since about 1992. And mRNA vaccines were developed for other diseases like Ebola before they were deployed against COVID-19. So it's not that radically new a technology. The vaccines consist of messenger RNA. That messenger RNA codes for a virus protein. It doesn't code for the entire virus. So if you get the Pfizer shot or the Moderna shot, you get injected with messenger RNA. Your cells will translate that messenger RNA into virus protein, and the virus protein stimulates the immune system just like the actual virus would. So your body thinks it's been attacked by the actual virus, and it makes antibodies against those virus proteins. And then the memory cells can last long enough that if you get attacked by the real virus in the future, your immune system can spool up very quickly and fight it off before it's had a chance to make you sick. The Johnson & Johnson vaccine is different. The Johnson & Johnson vaccine is actually a virus. It's a type of virus called an adenovirus. These are what adenoviruses look like. Each one of those little dots that I'm circling with the pen right now is a single protein. And the protein's packed together in a shape that it's actually the same shape as a 20-sided die. For those of you that have played role-playing games. There, I'm kind of drawing in the faces of the adenovirus capsid. So yeah, each one of these purple spots is a virus capsid. And the adenovirus that's used in Johnson & Johnson can't actually make you sick. What it can do is introduce DNA into your cells. And the DNA does not go into your nucleus permanently. It's not genetically engineering you, but the DNA is transcribed and translated into coronavirus protein. So again, your own cells make coronavirus protein. That triggers your immune system to respond. It triggers your B cells to make antibodies, but the adenovirus cannot actually replicate in your cells. So you don't get a permanent infection from the Johnson & Johnson vaccine. And again, we've known the outlines of how to do this for at least 20-odd years. It's not that radically new an idea to inject somebody with a crippled virus. Sounds kind of scary, but it's much better tested than some people realize. Incidentally, you can get your immune system to become sensitized to attack something that's not doing you any harm. So your immune system may freak out if you inhale pollen, which is about to start happening this time of year, right? Early spring, there's a lot of pollen in the air, and many of us are going to start sneezing. Or your immune system may freak out if you eat peanuts or strawberries or chocolate or what have you. That's allergies. An allergic reaction is the immune system overreacting to something that's not normally a pathogen and something that's not particularly harmful to you. And then finally, the immune system is brilliant in some respects and very dumb in others because it will attack anything that is not part of its own body. The immune system will attack deadly bacteria. It will also attack life-saving transplants, like this human heart that's being transplanted in this picture. This is why if you receive a transplant, you have to take for the rest of your life immunosuppressive drugs, which dial back the immune system's response. so that the transplanted organ is not attacked and ultimately weakened or even destroyed by your body's own immune system. So that's one of many reasons why all this stuff matters. And on that note, I will stop the recording here.